a de novo assembler that identifies non-chimeric contig sequences based on an underlying graph structure by Raquel Laniero and John Archer. Although artificially generated chimeric sequences can closely resemble expressed transcripts, patterns such as those between co-evolving sites and remapped read counts become obscured. This has a poorly quantified impact on downstream analysis, such as differential expression analysis. Here we present a de novo assembler that labels contig sequences produced with one of three levels that, depending on the underlying network complexity, reflects the level of expected chimerism within the data. The assembly process follows three steps: graph construction, cornerstone orientation, and graph classification. During graph construction, k-mers of length 40 are extracted from reads. The plot on the upper left shows that above k is equal to 20, increasing k-mer values makes little difference to k-mer coverage of the overall transcriptome. Edges are placed between k-mers, following which all unconnected graphs are removed. For the four species indicated, the number and sizes of these graphs are depicted in the bottom right plot. Ideally, there is one graph per gene family. For each graph, potential paths must be orientated. This is done by selecting a cornerstone node, as depicted in steps 1 to 5 in the figure. With the selection of the cornerstone node, starting and end points of paths can be determined, and these are placed into sets E1 and E2. Each graph is then classified according to one of three levels, the simplest being a graph with a single starting point, a single end point, and no internal cycles, such as that depicted in panel A. From such a graph, no chimeric paths are possible. Panel C shows a more complex graph of level 3 with 12 possible paths, most of which are likely to be chimeric. CStone is implemented in Java and is available from our SourceForge project page. An executable jar file, along with all source code and example data, can be downloaded. For demonstrating CStone's ability to assemble high-quality contigs, we used 4 simulated read datasets from fruit fly, leopard, rat, and canary, each containing 10 million read pairs, as well as 2 real datasets from fruit fly, each containing 30 million read pairs. These data are summarized in the table, along with the assembly times required for each, on 2 different platforms, Windows and Ubuntu. For the simulated datasets, reads were simulated evenly across the reference sequences present within the cDNA libraries available for each of the 4 species. This allowed us to remove the effect of random sequence error and expression differences from the assembly process. When simulated reads were mapped back to these references, a strong linear correlation, as expected, was observed. This is observed in the plot on the left. For the simulated datasets, the lengths of the contigs produced by CStone, RNA spades, and Trinity were compared to the lengths of the reference transcripts from which the reads were simulated. Top figure. When reads were remapped to these contigs, high R-squared values were obtained for the read counts versus contig length, indicating a good reflection of how the read data was simulated. If this was not the case, it would be an initial indication that the contigs were not reflecting the transcripts from which the reads were generated. When the lengths of each transcript are compared to its top matching contig identified using Megablast, significant linear correlations are observed for each assembler, confirming that the general length distributions previously shown are correctly reflecting the lengths of the reference transcript, top left. The bottom plot shows the lengths of the contigs against the aligned regions to the best matching reference transcript. For all three assemblers, the R-squared values indicate the contigs are aligning well across their entirety. Finally, each of the three assemblers was in high agreement in the number of reference transcripts represented by the contigs, with very few transcripts being uniquely represented by a single software. Similar results were observed for the real datasets mentioned within our poster. In conclusion, CStone does not offer an improvement on the overall quality of the assemblies produced by the other two well-established assemblers that it was tested against, but it does produce contigs comparable in quality, and it does add additional information to the outputs in relation to chimerism. This can greatly benefit the user during downstream data analysis, and it is easily adaptable to any de novo assembly tool. Thanks for viewing our poster and listening to this presentation.